I think we're live. Yeah. So we're here with the man, the legend, Ian Chen.
Hey everyone. Hold on, hold on. Can
From
I say the next few lines? Speed. No one
yes,
should watch this. No one should watch this.
no one, because I am unprepared. I am so unprepared. Uh,
Exactly.
so the idea is we have a rare species of. Hang on a second. It's not going to blur. We have a rare species of Australian canary. the The idea is to for me to take somebody who is not in the field of genetics particularly and to explain to them how junk DNA works. And so we wanted to pick somebody as a canary. So if the canary chokes and doesn't get it, then I've got to change the way I talk about it, right?
I
So,
think you really have to explain it exactly
oh,
how it was. It
they
was
used to
basically Speed that
they
said,
used to put a
"Hey,
canary in the coal mine, and the
yeah,
canary was the first to die of carbon monoxide.
yeah." So
So
basically, I'm the sacrificial lamb here. So I think it was last week when we spoke and you said, "Hey, what I need somebody to represent the average YouTuber, somebody particularly a creationist who doesn't understand the science, and if I can get that person to understand junk DNA, then I think I can be successful to get some of the creationists understanding. Hey, Ian, you look stupid enough. Could you be that
no,
canary?"
I didn't say
No,
that.
no. But I
No,
think that's what you meant, though. That's what you
no, I
meant.
said you were smart enough, but it wasn't your field, is what I said.
Yes, that's
So
true.
yeah. So and it was famously featured in the second season of The Boys on Amazon Prime. The Canary, you remember?
No,
Oh yeah,
uh,
and the
it is
second
super. No, I did see the second. What was it? A superhero or something?
uh the Canary was the guy who lost his girlfriend in the first season. I don't know the names of characters. I can't remember names.
Oh, the when that speedy guy smashed into her
Right. Yeah.
and
Okay.
oh,
That
the one
guy
with the
was
blonde girlfriend. Ah, yeah, 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 yeah.
he was the canary for the for Billy for William.
Yes,
Okay.
yes. He gets
And
put in
so
all the difficult situations.
goddamn
Okay,
Scott
at least I understand my role. Hey Scott, how you doing?
Scott is watching us. Damn, I told nobody should watch this.
Exactly. Don't watch it.
God, Scott always finds us almost instantly. That guy never sleeps. He just sits there
No,
on YouTube and waits for me to go live.
it's
It's
probably
like
not Australia.
what? Anyway, so my idea is like I look through my textbooks for junk DNA, and I looked at textbooks that were prior to 2010, and they never mentioned junk DNA at all. No evolution book that I could find. And then I look at textbooks from 2020, and they talk about it, but they only talk about it for a little paragraph. So my theory is that creationists are the ones that are making junk DNA popular again. They're the ones that are talking about it. And for some reason, Standing for Truth thinks that junk DNA is something that evolutionists require. That it's like the biggest thing for evolution. Yet I look at old books on biology and genetics and evolution, and nobody's talking about junk DNA. So if junk DNA is so important for evolution, why isn't it in any of the textbooks?
Hey, you know what'd be interesting? Do you know the history about junk DNA? Do you
A
like
little bit of it, yeah.
how does people like SFT etc. get this view that it is important to us? Like I don't understand that.
I
That's what I
think
mean.
the first person that mentioned it was uh, what is it? Ono?
Sus-
Oh,
yeah,
yeah,
Susuma Ono. Yeah,
Susumu Ono, I think it was, and
yeah.
he was the first person that mentioned it in a paper, and I think I have the paper somewhere, but it was sort of it's a surprise that so little of the DNA was involved in protein coding, and
Mm.
that's what they were surprised about, and they were like, well, what does all this other DNA do? But this was back in like the
But
50s,
look, hey,
60s,
so
and
can
70s.
I just sorry to interrupt you, but that's absolutely important. From what you just said there, Speed, you're saying that Susuma Ono was
Yeah,
actually when he says it's junk DNA, he's actually referring to non-protein coding genes, right?
right,
So at the very start, if he's the one that popularized the term, he's actually saying junk DNA is non-protein coding genes. So ergo, if we find that there are more genes that are coding, the term junk DNA is still correct because it's referring to non-coding genes. And if we,
right.
do you get what I mean? So it's not an incorrect term still.
No, Let's see. I do not have that paper handy. I know I have that paper, but I'm not sure where it is.
Did
But
you read
anyway,
it?
but the point was at that time they didn't know a whole lot about DNA. They just discovered the damn stuff. They didn't have a sequence, so it was something that they were actively exploring. And then by the 80s, by the even the end of the 70s, they had discovered long non-coding RNAs, and they discovered regulatory sections and introns. They discovered. So if you look at the genes here, we've got. Let's see, introns 5%. Okay, this is not. I've heard introns as much as 27%. So I'm wondering. Okay, we have repetitive DNA occupies 44%, unique non-coding, and then we have large segment duplications. We have simple. This is really odd. Okay, exons are only about 1.5%. They code for RNA, and then the introns are. Let me see here if I've got, like, okay, if you have a gene, and I have to make this a little
I want to make this
bigger.
big. Yeah.
Let me get rid of, I think I'm going to take myself off camera here too, because nobody wants to see this shit. Let's see. Okay, so can I make that even bigger, or is it going to, okay. So what we have here is a typical gene, running from, the edge of this blue thing here, all the way over to the edge of this orange thing here on the right. Okay, I don't know if you can see my cursor or not. You cannot see my cursor on the pictures, unfortunately. Okay.
I think, yeah, maybe.
Okay, but this is grossly misrepresented because the exons are usually a lot smaller than that. And so we have this intron space here, and the introns don't really do much. However, now, and this is where it gets complicated. When you start talking about junk DNA, you have to understand that the entire DNA molecule is a molecule. And molecules have the habit of having a chemical effect on everything else. So the entire DNA molecule is going to have some effect on the cell. So if it has some effect on the cell, that means that some of it's being transcribed, and I'll get into that a little bit. But okay, I'm getting distracted. First of all, these exons are spliced out, as you can see on this diagram here, and the introns are spliced out, and the exons remain, and that becomes an mRNA, or a protein coding mRNA. So that is the point I wanted to make, and I heard that.
From this pie chart here, hey, is it okay if I just interrupt with questions?
No, go ahead. That's why you're here.
When I look at this, it sounds as if at least 25% of the gene of the DNA does something, or coding, because you're saying you explain exons and introns, and I'm guessing when you have regulatory sequences that are around 20%, that's obviously that's functional. That's doing something important, right? Everything else is not important. So how do I marry this with when Gutsy Gibbon talks about 3% of the DNA is actually functional, etc., or was it Dr. Dan? So 25% seems much higher than the 3% number that they keep talking about. Or is it 3% just the exons and introns? Is that what they mean?
Yeah, the 3% are the protein coding part, the exons.
Yeah.
And actually introns represent quite a lot more, except that part of them are regulatory. Yeah. So it's really hard. It's really hard to say that any particular area is junk, but there is something we can do, and I'm going to try to share a different screen here and see if I can explain it. Let's see here. Application. There we go. And this is Michael's iPad. So let me see if I can.
That's what you use when you doodle. Hey, Nathan. I can see Nathan. Hi.
Oh, Nathan's here. Okay, so here we have the nucleus, right? And inside the nucleus, we've got our double-stranded DNA. And so the first thing that happens is called transcription. It gets transcribed. Okay. So transcript. Let me try to spell it right. Did I spell it right? It's early in the morning. I did.
Looks right.
So anyway, so from that DNA, we end up with this little stretch of, it's called like pre-RNA or something like that, and it's got exons and it's got introns in it, right?
Mm-hmm.
And then it goes down and it gets spliced out so that all we have, so this is I think it's called pre-RNA. There's a number of names for it, and it gets spliced out. So we get rid of those introns. We get rid of these parts right here, and we end up with just a big section of mRNA, which is messenger RNA. Okay?
Mm-hmm.
So it's important to understand transcription and translation, which are two different things. So that's one thing I want to teach you and teach everybody is the difference between these two terms, because you will find sometimes the creationists fuck them up.
So you're saying transcription is where something gets snipped, so it gets cut.
But it gets copied from the DNA up here.
Yeah.
Okay.
Yeah.
And it gets copied down into this pre-RNA. And then
Yep.
in the nucleus, it gets further processed here to get rid of the introns. So we want to get rid of the introns.
Mm-hmm.
Okay, like I showed you on that picture before. So
Yep.
we're going to end up with a very tiny, much smaller section that is right here that's called mRNA, which is messenger RNA.
And
Okay.
what does that do? What is the mRNA?
That is going to be exported out of this little portal. It's called a nuclear pore, and it's going to go out into the rest of the cell. So this is the rest of the cell. And out here, it is going to be translated. So now we translate it into a protein.
So translated basically means mRNA codes to a protein.
To a protein, right? So we have,
Right.
oh my goodness. So we have transcription, which is the first thing, and then we have translation. And so now this is famously where Maddox Beta Wolf talks about the ribosomes. So the ribosome does the translation to a protein. Okay. And that's like a little ball of stuff, and it goes through and it translates it. And then we end up with a finished product, which is a protein. So we have a protein right here that folds up and does something, and that is a protein. Okay. So does that make sense?
It does. So if I can just replay that back to you. Transcription is where you're basically chunking out a bit of DNA to get to an mRNA. So it's almost like a cutting down to an mRNA that does something. And translation is where the mRNA codes for a protein.
Yep.
So once a cut down of a DNA or cutting down from a DNA, and the translation is more whatever that mRNA output is, that mRNA, stumbling my words, that mRNA translates into or codes for a protein. That process is called translation.
Yep.
Okay. That's quite simple. Why don't creationists get it?
Well, they most of them have figured this part out, but they sometimes screw it up, which indicates that they don't have it quite really all that well. So in the nucleus over here, and then we have, this is called the cytosol, or the cell, the rest of the cell, the big bag of water in the cell. So this is a bag of water. Okay. We have, the cell, and we have the nucleus. So transcription happens in here, and then it goes out here, and translation happens out here. So this is translation to a protein. And making proteins is decidedly the most important thing that your DNA does. And this is transcription. So I should say just transcribe. And, oh shit, I can't do that with my finger. I thought I could do that with my finger, but okay, translate. So that's the important part. So this takes DNA and goes to messenger RNA. that goes out here, and that gets converted to a protein. Okay?
Hmm.
Okay. So that's a terminology: transcription, and translation.
Okay,
And
this is a little bit clearer now. So, because I know, and I used to be a little bit confused when Dr. Dan used to say, and when he talked about encode, he would say a lot of encode would be what he calls spurious transcription. In other
right.
words, yeah, in other words, I think what you're saying here is it cuts down. It does something that it snips the DNA, but it doesn't do anything part, whatever the mRNA or whatever it comes out of it doesn't do anything. So whilst ENCODE has shown that something has happened, which in this case is a transcription, there is no translation. In other words, it doesn't actually do anything to protein coding DNA. It doesn't code for protein at all.
Yep.
Yeah.
So
Okay, that makes sense. Yeah.
so we got a long thing of DNA here, and right here we have a little tiny gene, and here's another gene right here,
Hmm.
and here's another gene and another gene, and these are all genes, and inside those genes are introns and exons, right?
Hmm.
And those get translated out into proteins eventually, right?
Hmm.
So these are all about proteins. So what now what ENCODE is finding is that a whole bunch of sections here along this thing are transcribed into RNA of some kind.
Hmm.
Because when the let's say going back up here, okay, when this happens, these things are transcribed, and if it is a gene, it will get latched onto and it'll create an mRNA. But if it's not a gene, it will just create some other RNA. So it will be any old RNA. So that can happen. Okay.
Yeah.
And it is very so anyway, so if we look at this long thing here, we have the genes making mRNA, which is important for proteins,
Hmm.
but we also have thousands or tens of thousands of different RNAs, like small RNAs, microRNAs, long non-coding RNAs, so LNC RNAs. We have we have all these different RNAs that are being created that are all going into the nucleus or the cell, or they get out into the cell. And some of these things have a function that we know about, and some of them do not. Some of them are just seem to be spurious, like lots of them seem to be spurious. And what ENCODE found, ENCODE went down and they were able to use some these PCR techniques where you can take, oh my goodness, what just happened to my, I don't understand why that happened. Stop it. Stop it. Okay. So they could take one molecule and they multiply it using this PCR technique into, a million molecules. And then they measure it on a graph or on a blotter, so some device to read it. And they say, oh, so that little piece of RNA transcribed, like this little piece right here, we ended up with a piece of RNA from that. So they had very fine techniques to find everything that was chemically active. Okay.
Hmm.
So So anyway, what Dr. Dan is saying, and he rightly so, is that this was one molecule is probably not going to make a difference in the cell. That is called a spurious translation. Okay.
Which is what percentage of the DNA that does that?
We don't know yet. We're guessing. Pretty sure that.
Something less than 75%, I'm guessing. Given
Uh,
25%, sounds like we're doing something.
Actually, yeah, definitely less than that. So more like about I'm thinking, well, okay, about 80% of it they found out does get transcribed somewhere.
Yeah.
So they've said as high as 80%. They've gone down to about 71 or 72.
Hmm.
And you'll find that ENCODE will no longer use the word functional, because that is the whole problem here. Is in the beginning they said that this one molecule was functional.
Yeah. Okay.
Well,
So you're basically saying, depending on the studies, between 71% to 80% of the DNA is spurious transcription.
well, Yes and no. It's more complicated than that. It's much more complicated than that.
You're hurting my
So,
head.
and that's the problem. Is because they miss all the nuances, right?
Mm,
So in this entire strand of DNA, we have all this stuff. Some of this stuff is like one molecule, sometimes 11 molecules, sometimes 100,000 molecules are made from, different pieces of DNA. And so, but if we have one molecule, the chances are it isn't going to do anything. We don't know exactly how many because they can't count how many molecules.
mhm,
We just know, well, they can. They get these little bar charts where they'll tell you that, well, that's one molecule, and this one over here is 11 molecules. So they can kind of tell you how much is being translated.
mhm.
But the point is that
Transcribed. You mean?
this is on a scale.
Yeah.
So we have a scale here, of okay, really functional, like really important in the cell, like collagen, which is, the most prominent protein, I think, in your body. So this is really important down to does nothing at all, has no effect on you at all. So you won't, the only effect it'll have on you is you might want to take another bite out of your sandwich because you created something you didn't need, right? So we have this range in between from 0 to 100 of how useful it is to the cell.
Mm,
Okay. So when they talk about junk DNA, they're trying to draw a line in the sand here, which you cannot draw. Does that make sense?
yep.
So we have the cell, which is a big bag of garbage, basically, of a big bag of mud. I call it a ball of mud, which has all kinds of biochemically active chemicals in it.
Mhm,
And inside of that ball of mud, there's everything from 0 to 100 as to how important it is. Now, whether or not this ball of mud survives and is capable of dividing into another cell depends on what exactly the composition is of, inside the ball of mud. For instance, if we put a toxin in here, the cell will die. If the cell gets a signal that says die, there are death signals that one cell will send to another. You end up with something called apoptosis, which is
mhm.
cell death.
Yeah.
Okay.
Yeah.
If
I
we
heard
get a
about.
signal in here that says grow like a motherfucker, we end up with cancer. Okay. So it depends on the composition of this cell as to whether the cell becomes cancer, whether it's a normal, healthy cell, whether it's a cell that's going to die or somewhere in between. And so you have, 100 trillion of these in your body, and they're dying left and right. There's mutations, there's problems. So you can see how complicated this is getting.
Hmm. Yeah.
Okay. So do we need to review anything? So
No, I do want to kind of say, so it does present a little bit, at least the way I understand it now, a little bit of a problem to evolution in that kind of creationists argue that even, if you go up, can you go back up to your chart?
this one?
Even, yeah, even if it's one, let's say
Right.
even if it's one, couldn't they argue it has an effect? It's just really, really slight. And if you remove enough of these ones, it's going to be detrimental, ergo genetic entropy, blah, 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 blah. So um, as long as most of it is zero, then evolutionists can kind of say, hey, a lot of it is definitely junk because it absolutely does nothing. But even at one, kind of creationists say, look, it might do something, but it's very slight, and it's a slight benefit it's conferring. So if you remove it, maybe you can't see it, but it's still a slight benefit it's conferring. Can't they argue that?
They could, but they would be wrong.
Yeah.
Uh, if you have your, let's see here,
So you're
your
almost saying that there needs to be a minimum. So that minimum is not one. That minimum might be 50. So anything
right.
under 50, it absolutely, if it does something, it just makes no impact at all. So there is that minimum level, and we just don't know what that is at the moment. So it's very hard to push back on a creationist. I'm guessing, is that it? Am I
Yeah,
understanding that?
here's your car battery. Okay.
Yeah.
And What the hell happened to my drawing? Okay, your car battery, and you're running your car, and you got the radio on, you got the headlights on, you got tail lights on, and all that stuff. And suppose you turn the blinker on for just one blink. Okay, that's one molecule. That's all it's going to do. That isn't going to make your car battery dead.
Yeah.
Cells are extremely durable. If they were not durable, none of us would ever live longer than the first few hours of our lives after conception, right? So it's very, very durable. So this basically this whole percentage game inside the cell, when you have something that is like 0.001%, which is probably overestimating one molecule, it is not going to shut your car down. It's not going to kill your battery if you turn that blinker on just once, right? You're not going to have a dead battery. So your cell is going to live if you have all these one molecule, 1, 5, 10 spurious transmissions, because you have, I'm not sure how many molecules are in the cell, but 100 million, maybe 30 million, 100 million, I'm guessing. So one molecule isn't going to do a lot. It's going to really work. Just refer to me. Oh my God, Andreas Brahmer. Andy's here. Now he's an interesting person. Okay, he says transcription activity and function are distinct. There are some genes that encode RNA sequences. However, genes code for RNA sequences that have no apparent function. Yes, no apparent function. So anyway, the point is that if they argue that, they would be wrong. One molecule is not going to have any effect on the cell at all. You're not going to notice it. You turn your blinker on, your headlights aren't going to dim, right? Yeah. Unless something's really wrong with your car. So the thing is, we are these balls of mud work on statistics entirely. They statistically work. So it's either going to be alive or it's going to be dead. Statistics. So this is a statistical game, and that's what they miss. They want to see everything as like a factory or a machine. Well, if you have a factory, this perfect factory, right, that's spitting out cars. If you have a gear in that factory, I can't draw gears, right, but that's a gear. If you have a gear turning another gear and you throw a wrench in that gear, it's going to shut down the factory, right? Okay, the cell is not like that. You can throw a lot of wrenches in this ball of mud before you're going to affect it. In fact, that's how life works. If you go outside and you don't put your jacket on, you go get the mail, you don't die on the way to the mailbox. Your body turns up its heat. Your cells compensate. So it's a statistical game is what I want to get at. So the important thing is to remember a ball of mud that works on statistics. Okay.
Yeah. Hey, a question did kind of come to mind. Okay. So I'm trying to think this through in terms of how did junk DNA evolve? I guess. So I'm guessing with evolution, over time, as you kind of evolve, there would be previous functions, etc., that used to do something and potentially it doesn't, and is junk DNA the kind of remnants of that? Like how did junk DNA evolve?
Okay.
How did we get to be 75% of our DNA that does nothing? Like how did that come about?
That's the interesting part. Now in bacteria or protists, they have much less. Usually, well, no, that's a damn lie. That's a damn lie. There are chromosome duplications. So chromosome duplications. There are sections of DNA that get duplicated. There are endogenous retroviruses that get in there and they multiply themselves and they keep extending the DNA so it's bigger and bigger and bigger. They're in like mice, there are chromosome fusions where there are 92 different chromosomal patterns in the house mouse.
Okay. So you're saying junk DNA seems to come from where the DNA
accident
grows. Yeah. So it's where its chromosome gets duplicated, DNA gets duplicated, ERVs get inserted into DNA. So
Right.
junk DNA is essentially what happens when the DNA grows through these methodologies.
Yep.
And because they don't do anything, you now suddenly have this stretch of DNA that's now non-functional to the survival of the cell,
Yep.
essentially. Okay,
And
gotcha.
there are, let's see, what are the other methods? And what you said, suppose there is a gene here that is no longer, for some reason, because of the change over time, this gene is no longer useful, then that would also become a little bit of junk.
Okay, so just if I kind of extend what you've just told me, does that, can I infer that the further back in time we go, the less and less your cell is junk? So something like a bacteria or whatnot would had you presumably have less junk DNA, and something that's more evolved or more complicated, in other words, it's that has more time to evolve, that would probably have more and more junk DNA. Can I infer that? Does that kind of make sense?
Yeah, but not necessarily.
Hmm.
Not all the time. Sometimes there are like you could lose a chromosome, or there can be like a massive deletion of an area, but that is not as common because usually the deletion is going to kill you because the
Okay.
deletion is going to have.
So, so tiktaalik, okay, because we came from fishes, became tetrapods, etc. So let's pick an ancestor way back in time, a tiktaalik, let's say, or something similar. Would tiktaalik or that something similar's genome have less junk DNA than us? Because obviously over the 200 odd million years there would be these duplication events, etc. And then presumably from what you just told me, the junk DNA proportion would be growing and growing over time.
Yeah, it's so almost for certain. The further back you go in a lineage, you got these branches, that come out, and basically a phylogeny that keeps branching out. So as you went back, because of ERVs, you would almost certainly get less and less DNA as you went back.
Yeah.
Uh, but even that is something that you can't be sure of. I'm sure there are cases where there was like more DNA back here, but I'm trying to think. To lose DNA is a little harder than it is to gain it. To gain it is, we end up with this big mess of DNA out here, and as we do, the animals they could get more complex, they could get simpler. There's no rules. Every time you find a rule, you are going to find an exception in genetics. Okay, and that's what the creationists will find a paper with these exceptions, and they will try to say that the exception is the rule, and there are no there is no rule that doesn't have a violation. So as soon as I found a section of DNA and I said, this section of DNA here is completely junk, a paper is going to come out to say, oh no, this little section right here creates a little tiny RNA that now has some effect. Like for
Yeah.
instance, standing for truth, ERVs.
Mhm.
He found an ERV or two or six that is now functional because it's been around long enough, and we're going to get to that part.
And for it to kind of mutate, I get that. Hey, so firstly, thank you for explaining all this stuff to me. I didn't quite actually appreciate or understand it as I did prior to this stream, but I wonder why creationists think junk DNA is a problem. Now, I understand that it's a question around obviously if most of your DNA is non-functional or non-coding, then you're not perfect, right? So I get why they have a problem with junk DNA. But the way you've explained it to me doesn't can't you kind of interpret that in a really twisted way to kind of support genetic entropy? So if we're kind of saying over time with DNA, I mean, forget the time discussion aside, forget the fact it's billions of years, right? Over time, as an organism evolve, you're going to get more and more duplication events, etc. And a lot of those events might not end up doing anything and end up being part of your junk DNA percentage. Couldn't a creationist, I mean, I just don't understand why they think it's a problem because they can argue that this is genetic entropy. Over time, because man's getting worse, you're going to get more and more of your DNA that essentially does nothing. In other words, becomes wasted. Can't they argue it that way? Like, why do they think junk DNA is a problem?
Okay. For one thing, they have the idea that it is perfectly designed by God.
Yep.
With
But they
Adam
also
and
have
Eve.
genetic entropy, right? So
Right. And then they want to say that if anything mutates, it's negative. Okay. So they want to say that this is deleterious, always deleterious.
Yep.
There are no neutral mutations. So we have perfection in the beginning, and then they want to say that all these mutations are happening, yet life goes on. But they think it's going to end in this big apocalypse, like within 300 years, right? So apocalypse. So they think this is going to happen. So they think we're heading. But the thing is, they want to say that it's all functional, and yet they want to say that it all mutates. Well, those two things are contradictory, okay? Because if it's all functional, we would not be alive. In fact, the thing is, when you're born, this is you as a little baby, right? A little squalling baby or a little fetus. Fetuses look like that. So you go from fetus to baby, and this is 9 months. Okay, and then you go out here, and now you end up with Big Ian, my canary. At whatever 100+ years out here, and around 100 years. The thing is, you would die right here if we had 100% functional DNA, because over your somatic life, you have mutations every time your cells divide. And I mean, in the beginning, in this first 9 months, your cells are dividing like fucking crazy. They're constantly dividing, and there's constant mutations. And if all of the DNA was functional, you would never get born. So the fact that there is life at all means that they are contradicting themselves by saying that we have 100% functionality. There's no way for 100% functionality to work.
Andres has an interesting question on the side
Yeah.
chat. Yeah, basically, I think he's talking about the ERV that you're talking about. Like, what proportion of that?
Yeah, we're gonna get to that, but we have to get to something really important here. That and I hope I haven't. You see, when you have too many concepts, sometimes we have to review. So we have the transcription, translation.
Yeah. And I'm following so far. It's good.
We have statistics, the ball of mud. You understand that life. Jeff Goldblum, life finds a way. These cells find a way. So you are very, very durable. I mean, you can go out, you can mud wrestle with a creationist, or ENCODE refers to that as mud wrestling with pigs, though I would never call them that. That's just ENCODE. You know about that, right?
If you wrestle with a pig, only the pig's happy or something. You both get dirty. Is that what you?
Yeah, that one.
Yeah.
That I actually got from a high up in the ENCODE project.
They
don't like creationists.
No. They certainly give them a lot of fodder. That's for sure.
Yeah, they give them a lot of fun. Yeah, and I confronted them about that, and I said, "Why are you giving these guys so much shit?" And that was his response to me. But anyway, so where was I going? So anyway, okay, we reviewed that. Now here's the thing. Now, if we have a strand of DNA, okay, and I look at your DNA and I look at my DNA, and we have two strands and we compare them, we're gonna find just a ton of differences between you and me. We're going to find lots of alleles that are different. And this is heterozygosity. Well, not heterozygosity, but this is variation within a population. So between me and you, there's a lot of this DNA that is called non-conserved.
Yeah, I'd probably say, just based on what I know, that it comes down to the definition of what you mean by a lot, because I'm pretty sure when we compare our whole genome, it's probably something like 99.999% similar.
Yeah. So between me and you, we'll end up with like 0.1% maybe difference, right?
Yeah.
So that is the non-conserved part that makes us both human, yet we're both alive, even though we have all these differences, right? But now if we go down and we look at a chimpanzee, we'll end up with 98%.
3% or
Or,
3%.
or so we have 2% that's non-conserved between us and a chimp. Okay.
So can you explain again? What do you mean by non-conserved?
Okay. That means that it doesn't make any difference between, we're both humans. Okay. We look a little bit different, but we're both very much the same thing. And that's due to this 0.1% that is non-conserved. It doesn't make a difference to the fact that you and I are both viable living humans of the same species. Okay. So if you go down to a chimpanzee, you'll find a 2% difference. So 2% of this DNA, the creationists want to tell you it's 14%, but we could get to that later. But anyway, so it means that much of the DNA can mutate and not really make a big difference. Okay. And now if you go down and you look at a mouse for some particular gene, you will find that only like 52% is identical and conserved. Okay. So that means that 48% is, we could call it junk. Because
I do have a question here. Where do you draw the line between species? So obviously 0.1% is not the line because you and I are of the same species.
right.
Obviously 2% is way past the line because humans and chimps are different species.
Yeah.
At what percentage point is it then you kind of go, I'm just trying to get a, how Ken Hovind and whatever that he always goes, you guys kind of define what a species is and it always changes, etc. Is it possible to define a species by the genetic variation? Can we say at 0.2, 0.38, or whatever the number is, can we say anything above that it would likely be a new species?
No, you never want to use the word species when you're talking about genetics.
Yeah. I mean, but there is a gradient. Obviously, 2% is way past that gradient because a chimp and a human with that difference is clearly a different species.
I bet you could find an exception to that. There might be
rules with exception that you mentioned previously.
right. There might be one thing here that if you change it, you'll end up with an entirely different species. One little tiny bit of patch of DNA, and so you'd have like 99.9% identical, yet we have a different species. I don't know if we found that in bugs or anything, but again, those rules do hardly ever apply. There's always those exceptions. So very, very, very hard to talk about.
It is difficult because it isn't like Neanderthals something like 0.3% different, and clearly we're a different species, but we can still reproduce.
Right. Yeah. So there again, there are exceptions all over the place, and you can't find any rules. So the problem, but the thing is, when we look at these DNA sequences and we can all look at them, we find these okay, if you're looking at like mitochondrial DNA, mitochondria they're very, very concise little machines, and they don't have a whole lot of fluff, a lot of room. So they're highly conserved, so if you have a gene, like for a protein right here, okay, you'll find that between a mouse and a chimp, about 52% of that DNA is highly conserved. But we can't mutate that because it'll end up with a different kind of protein. But we can mutate 48% of it and end up with a viable protein that does something. And here's where it just all gets crazy and goes to hell. In that sometimes one single amino acid is going to make that protein not do what it does, right? And we may have two copies of that gene out there somewhere, and this one is going to keep the cell alive, okay? And this one is kind of mutated, so it's not going to do anything, but it is chemically active. So as we move along through different environments and we reproduce, this thing could gain a function at some point. So it has a function now. So now it becomes part of a new kind of species. Okay.
Hmm.
because it's gained a function. So I mean, this is where, yeah, let's see, what is the takeaway here is that we should be confused at the end of this when we talk about junk DNA, because junk DNA
You're
doesn't
doing a
make
great job.
any fucking sense, right?
Yeah.
So if we go any further, but this thing, so the thing, instead of so let's say we have junk DNA, which the creationists like to talk about, that is bullshit. What we want to talk about is conserved and non-conserved.
Hmm.
What stays the same between me and you? What stays the same between us and chimps, which are similar? So non-conserved versus conserved. What stays this? What makes a difference? Basically, is what we're looking for. So if we go from me to you, some of those things are going to make a difference, and we're going to look a little bit different, right? We're
Hmm.
different people. We have slightly different brain wiring, but we're still pretty much viable human beings.
Hmm.
And
Hope so.
you know, but it's fuzzy as to what we're calling human or what we're not. Because when you get down to chimps and then you go down to mice, we're going to get like a different phenotype, and we're going to have less and less DNA, but we're all mammals. So if you look at mammals, you're going to ask what is conserved in all mammals, and we're going to be able to tell what's similar. If we look at primates, we're going to ask what is conserved to make a primate. If we look at human beings, me and you are going to ask what is conserved, what is non-conserved.
If you can just hang on a second, Speed, I just want to answer, just make a comment to Andre's comment around how he says there are at least 26 ways to define species. I guess,
Yeah.
Andre, this is my problem with, people like Ken, etc., where they kind of point to the fact that even evolutionists can't define a species. So ergo, it's okay that creationists cannot define what a kind is. And hence my question is Speed to try and kind of flesh out, is there a better way to define species so we can kind of negate those arguments from creationists? And it just sounds like it's just difficult. I think that's what you're saying, Speed. It's difficult to define a species.
I don't know who this Andreas Brommer is. He's probably some sort of spammer or creationist, or I don't really know what he is, but
No, he's asking interesting questions. So
he is asking interesting questions. But I don't know if I should I don't know. Uh, I don't trust anybody named Andreas Brommer. Anyway,
Andres.
Andre. Oh, Andre the Giant. Okay. So anyway, so the takeaway from this is, how in the fuck can we even use the word junk? I mean, it doesn't make any sense to even discuss it. And yet the creationists are constantly saying that is the most important thing in evolution. Well, that's bullshit. So, okay, we have this non-conserved part. Now, the other part is that, so we have a whole lot of DNA that makes a difference right here. And then we have a whole lot of DNA that is kind of non-conserved that can change. And this is the DNA that is going to give us new species eventually and new biochemical pathways. So this stuff is like mutating, and it's going to have some effect. It's going to make us maybe a different species, or make us look different, make my skin color different, whatever. But for the most part, we're still going to be alive. But then every now and then, there's going to be a change, like there's going to be a delta or a change that amounts in a new species, or some new function, and we're not even going to see that happen. Like if we looked out in the woods right now and looked at all the insects, there's a population of beetles right here, right, that has gone along, and then all of a sudden it branched out into a slightly different population with a different biochemical pathway. So this is called the neutral DNA. It's a DNA that doesn't kill you, and it doesn't make too big a difference all at once, but eventually it will make a difference in a species. So this is where evolution happens, is in this neutral area. So we have all this, instead of junks, we want to call it, like if we had, We got a factory. Okay, we got a big factory, and we're building shit, and we have all these machines that we're using every day. But in the back room, we have all these parts of machines, that are just floating around back there. And every now and then we drag one out and we use it. And that is how evolution happens.
Mm.
Every now and then, one of those little pieces in the back room, that like we may take one of these pieces and we may put it in the back room because we don't need it anymore. Or we may take one and we make a copy of it and put it in the back room. So now we have a copy, and now we have two of them. But then over time, it starts to like modify. Here's where the factory analogy breaks down. If you have a hammer in the back room, if it's sitting in the back room and nobody's using it, your hammer does not change into a hoe or into a rake. But in fucking life, it does. It changes when it's in the back room, when it's in that neutral area. So this is where the machinery of evolution happens. And because it's a
This is
big
one of the.
hole. Go ahead.
Yeah, I was going to say this is one of the most frustrating things I hear from creationists all the time. So, if you go back to where you had that kind of factory drawing, so when you delete something, they're just going to say, "Oh, that's not new information. All we did was we deleted what was already there." When you're copying something, whether gene duplication, whatever, they'll just argue and say, "Well, that's not new information because you just copied something that's already there." And
Right.
so because they say that, it doesn't matter whether the second copy then changes, a C to a G or whatever to become something new, because they'll just base it on well, the copy was just a copy. It's not new information anyway. You've just copied what was already there. Now, this is why I think the creationists cannot grasp the concept of new information, and they can't grasp the concept of it doing something different because they see any deletion as a deletion, therefore losing information, and anything as a copy is copying something that was already there. Ergo, it's not new information because it was already there.
Right.
So that's one of the things I find super frustrating with creation is they just can't understand, new traits. I think they look at it black and white. They don't get that even if it's a new trait, that's new information.
So this is our hammer copy. So what they're looking for and what
Sorry, it's a Bremer copy.
a Bremer copy. Yeah.
Yeah,
You got
Andres Bremer.
a
I'm gonna
Bremer
say it
copy.
correctly. Yeah.
Okay, so we have the hammer copy, and it slowly mutates into what the hell could we mutate that into? Like a wedge. Okay, mutates into a wedge because it loses its handle and a bunch of other shit. So now it's a wedge. Okay, so now it's a new thing, and what they're looking for is they're looking for instances where a hammer turns into a wedge and then gets re-employed back in the factory so that now we're using it. So they're looking at those things. And they have been shown those things in a couple of cases. But the problem with that is the world is really big. And we know of 1.3 million species. We think there are 10 to 30 million species out there. And we can only so far we've kept track of the zebrafish, the fruit fly, human beings. We have like maybe 5 species that we really know a lot about. In order to find that hammer turning into a wedge, we would have to get the genomes of every goddamn insect in the forest, every prokaryote, every protista, everything, keep track of them for 100 years. And then we would find all kinds of examples of this happening. We would find new biochemical pathways. So what they're bitching about is that we are human beings and we don't have enough of us to keep track of what's happening in life. There are some cases that they have missed, like ApoA1 Milano, that little protein.
What's that one again?
That's the protein in it.
Chen talks about it all the time, but I just don't know what it is.
Okay. ApoA1 Milano is a protein. It's ApoA1. And ApoA1 is a protein that's important in collecting cholesterol, getting rid of the bad cholesterol out of your blood. And what it does is it's got a protein that attaches to one or maybe 3 other proteins they kind of glob together, and they form this complex outside of your cell that gets rid of cholesterol, right? Well, ApoA1 Milano modified one, I think it was an arginine to a cysteine, I think it was, or something like that. And what happened is now these things cannot form threes, they can only form twos and fours because cysteine, like it's a sticky amino acid, so it sticks two of them together. So they form either twos or fours. They can't form threes and sixes and whatever. And this turns out to be like 40% more effective at every function. So this is an actual mutation that made an improvement in human beings. But they'll say, "Oh, but that's not a new limb," you know. They'll say,
What they'll say is it just copied another one of those circles or something that you've just drawn.
"Yeah, yeah." But they quit talking about this because I showed them where they were wrong. They were saying that, "Oh, it lost function because now it doesn't work as an antioxidant." Well, they found out, I think, that it's like 300% more effective as an antioxidant. So this modification, this one nucleotide
When you
modification,
say they, who do you mean? Like who's not using it? Who did you educate?
who did I refer to as they?
You said they stopped using it once you explained it to them.
Oh, the creationists.
Uh,
Back in like March or April, I had a thing with standing, and we
so
were
it was
talking
the
about
brain trust.
ApoA1.
It was the brain trust you've educated.
The
Gotcha.
brain trust. Yeah, they don't talk about it anymore because I showed them papers where they were wrong, and Raw Matt had a bunch of references to papers that didn't make any sense at all, didn't have anything to do with ApoA1. But ApoA1, this is Milano. Milano. It's a small town in Italy that's not named Milano, but I don't know why they call it. I think because the University of Milano or something found it. But it's just a small population where they found this modification. And these people are eating eggs and meat and they're living to 100 years old. And they're like, what in the hell? All of a sudden they don't get heart attacks, you know?
Yeah. Hey, so you could almost argue that if you were successful getting the brain trust to stop using it, that at least they'd know Ken Hovind. I've seen Ken Hovind being corrected hundreds of times and still use the same wrong arguments all the
Yeah,
time.
he
So
probably uses this. I don't know if it was ever one of his.
Ken would not understand a word you just said. No, he wouldn't get any of this.
What's that?
Ken, did you say Ken would you be using this? No, Ken would not understand a word you're saying.
No, he wouldn't understand a word of it. So
He'll think Milano is some sort of fashion brand.
yeah, right. Anyway,
Shirt fashion brand.
so what is the takeaway? The takeaway is that junk is bullshit. You know, if they start talking about junk DNA, what is an example of what they're doing? They're trying to, like, is a red herring? Would it be a red herring? It's like, and we go after it, you know, like Dr. Dan will defend, you know, by telling about the spurious transmission. But the thing is, they aren't even wrong about this. I mean, it's like they will talk about this junk DNA like it's the most important thing to evolution ever.
Who's that famous evolutionist that had the saying that basically said you're not even wrong?
Yeah, I can't remember who that is.
It was
Yeah,
an evolutionist or a physicist. Yeah, I can't remember. It was a very popular saying where you're not even wrong.
yeah, I
You're
can't
so
remember
far
who
off,
that is.
you're not even wrong.
Not even wrong. Yeah, yeah. So when they talk about, you know, the thing is when they talk about junk, you could say, yeah, there's some junk, but no, it's not really junk. But you know, and I mean, it's just it's a distraction that they believe in, that they want to make big noises about. Junk DNA has been overturned. It's like, and I could say, yeah, I agree with you 100%, but it's more complicated than that. I mean, so if you're a little confused by all of this, well, we can make you more confused.
No, I'm getting the concept, but
Yeah,
I guess what's difficult is there doesn't seem to be any hard and fast rules, and that's why the creationists can hide behind this because there's no hard and fast definitions they can hide behind. basically, what they're doing now, where they confuse everything.
Yeah, so here we have exons, and we have introns, and we have exons. Okay. So when there's mutations, there might be some mutations here in the exon, which would change the protein, but there may also be even more mutations here in the intron, which means the intron is less conserved than the exon, right? So the intron has a little more wiggle room than the exon. But then we found out that some of these exons actually have small sections that get copied into RNAs that actually have a function. And then inside the exon, just to get this thing to splice properly, there are a couple of signals. There's like a hinge point and a couple other signals in the intron, and we need those because if you don't have those, it's not going to splice, right? So this part of the intron is conserved, okay?
Yeah.
But not all of it is, but a lot of it is. And so how the hell are you going to say that any of that is junk? And as soon as you say that this part is junk, well, you'll get a paper out there and say, oh, no, we found out that this copies into a little miRNA that causes a liver cell to do X. So it's very, very complicated, but there are definitely huge sections of the DNA that do not make a difference. We can mutate the hell out of it.
So if I go back to you at the very, very beginning, this 1.5% exon introns with 5%, and that green stuff that you got in there, that's what you're calling the 20% regulatory.
Yeah.
Yeah. Okay.
Yeah.
Good.
We'll go back to that.
Yeah.
Let me go back to that window.
Yeah.
Yeah.
That one there. Regulatory sequences. That's where you had the green lines.
Yep, but they're going to make the regulatory the purple part. There's the regulatory part.
Yeah.
Unique non-coding. I'm not sure what they mean by that. I just pulled this this illustration up this morning, so I haven't seen the paper. But this whole part over here, these repetitive DNAs that include all the ERVs and all that stuff, there are huge sections there that don't make any difference at all as to how they mutate. If they were to mutate, they don't make any difference at all. But it gets worse. Okay. So you've got this huge piece of DNA, and you got, like this is functional.
Hey, can I just ask a question, Speed? Sorry,
Sure.
I too tend to look at this in the context of, things that I hear that creationists kind of say, right? So one of the things that SFT keeps on saying is, how do we know it doesn't do anything? Because he has said things like, the gold standard is gene knockout test, etc., etc. We have not done it for anything more than 1% of the human genome, and etc., etc. So his question, or the creationists would argue, how do we know it doesn't do anything? How do we know
I think
that?
in Drosophila, they knocked out everything except for 12%, and the thing still worked.
Okay, so for the fly, it's all right, but for humans,
For the mouse, they've knocked out a ton of shit. For humans, we can't do knockouts, so we don't know.
Probably not ethical. So what we're saying is that we've done it for some of the more simplest species. Probably simplest not the right word, and therefore it kind of makes sense that this trend, or what we're kind of seeing, is probably correct for humans as well.
Right. So okay, so say this is all ERVs here, and one of them right here has a function, and this one has a function. So they find just a few because these have been around for millions of years, and they're part of that. They're part of that junk toolkit out here that we were talking about out here. So they're part of that neutral DNA. They are going to eventually find something to do if we keep them around long enough. But here's the thing. Okay, this is a bead on a rope. If you imagine beads on a rope, so we have these DNA beads on a rope. The length of the rope might be important. So just the length of it doesn't matter what the rope's made of. What matters is that we can hook the rope from one place to another, because inside of the nucleus, the DNA is arranged, like beads on a rope, and it's arranged in areas. and it makes a difference how long that rope is. So as we evolve and add ERVs, so sometimes when your say your brother ends up with a duplicated endogenous retrovirus, and he ends up with one or two too many duplications, he can develop Alzheimer's disease or some other disease because the length of the rope makes a difference. So are you going to call it junk or not? You know, it doesn't matter what the rope is made of, but it does matter that you have the rope. So that's another complication that we can add to this mix. And another, so I think what I'm trying to do here is confuse the hell out of you, Ian.
No, I'm getting the feeling that the DNA molecule can be read in so many different ways. It's not just the like it's not just the letters on a page in a book. It's
Right.
also how big and thick the book is. It's like every part of the DNA is, you know, 3 or 4 dimensional, and that it can be read in different ways. And that's essentially what you're saying. It's not as simple as just reading a code in a 4-letter way. It's also the length of the code and all that. It all does something. So it's more complicated.
Yeah,
That's my big take out of what you're saying. It's complex. It's hard.
and then Otangelo will say, "Oh my God, it is so complicated. This language is so complicated, we can't even read it." And but it's not a goddamn language.
You can't read it now, but if you go back in time, it's less and less complicated, right? So it makes perfect sense to me that the difficulty is just a function of time
Right.
and evolution, basically.
It's a ball of mud. It's not a language. There are so many, you know, like we have there are no rules. A language would have rules. Okay. Every goddamn rule has an exception in DNA. It's going to have hundreds of exceptions. It is going to have backwards and forwards exceptions. It's going to have 1 and 2 and 3 and 4 dimensional exceptions. So, I mean, it's just this constant, as soon as we find one mechanism, like if you have a gene, there's a little signal up here basically that tells you whether the gene is going to be on or off. They are finding hundreds of different ways that can happen. If you have a gene and we say that AUG is the beginning of the coding part for the protein, which is methionine, they will find an exception to that just as soon as they found it. So every single rule is going to have dozens of exceptions because life is a ball of mud. It's a fascinating ball of mud because it sticks around, right? But I mean, you can't. So anytime they try to lead us down this pathway, yeah, here we go. So I think he said it's complicated.
Yeah, bioswooning.
Oh, bioswooning. Yeah, bioswooning. So I mean, so at the end of this, you should know that when they use the word junk DNA, that they are going. The next thing they are going to say is going to be a lie. Okay. Because anything you can say about junk DNA, one way or the other, is going to be a lie because it's bullshit. I don't know. Have I done any good here?
Oh no, I definitely understand more than I did before your stream.
Good.
So thank you there. But I think you've also highlighted there's also a lot of stuff that I have yet to understand.
Me
So
too.
I understand when you say it's, you know, it's not just the letters within the DNA, it's the length of the DNA. So I get all that stuff though, but that's where my knowledge stops. The
Yeah.
detail that's behind that, that's where you're going to lose me.
Yeah, we probably shouldn't go any further with this.
But the
Right now.
canary's not dead. The canary's not dead. Just
I haven't killed the canary. Good.
I haven't killed the canary yet.
Yep. Well, your
If you go any
question.
further, you might. Pardon?
Your questions help guide me because if I just started talking about this, it would just become a confusing ball of mud.
How did you learn all this shit? I mean, that's, you know, you don't have you've not studied it formally. So I'm impressed that you've picked all this stuff up. But I guess it's just interest, right? You're interested in the topic.
Well, yeah, I keep studying it. I actually read the textbooks. I actually spend time like a
But
student.
according to Stinky Snake, you don't read
What?
any books.
Yeah, right. Yeah. But I just keep chipping away at it. But the problem is like I will set out to understand everything there is to know about transcription. And by the time my first cup of coffee is gone, I realize, holy fuck, I don't know anything about transcription. I'm never going to, because there are too many things to learn. As soon as I learn one thing, I find another thing and another thing. And how do I learn all this stuff? You don't. Nobody is going to learn all of this stuff. It's just a continual. Uh, you continually expand, I guess, your overall intuition about what's going on. But as soon as you figure something out, like if I say miRNA is only used in the cytosol, well, yesterday I found out no, it actually works in transcription as well as translation.
Yeah.
So I found an exception to the rule. So you don't learn it all. So,
You know where I think the problem is. The problem is, and it's I think it's the evolutionists that have not done any favors for themselves. So when you sit down and show me this chart and explain it to me, you're being very precise in your language. The problem is when whoever came out with junk DNA, whether it's this Ono person or whatnot, because it wasn't used in a precise manner, creationists jump on that and basically twist it around to kind of make it sound like, we don't know what we're talking about. Whereas junk DNA it's almost a catch-all for a lot of these parts of this chart. Do you get what I mean? We're not doing ourselves any favors because we're not being precise in our language, and that
right.
gives fodder to creationists to kind of argue how evolution is wrong because they don't take the time to understand the language either, and they just take the very key high-level kind of headings and then basically use that to attack evolutionists because the headings itself is not a precise description of the science.
Yeah, because Ono wanted to say something, wanted to make a point, and use
Yeah.
the word junk DNA, and it became it became an anchor around our ankles,
Yeah.
for forever.
Yeah.
Because of but yet Ono was just saying one thing: is like, why is there all this? Why is there so little protein coding DNA? That's what they were talking about, and why we have been answering that question for 50 years.
Yeah.
And it just keeps getting. Let's see. Scott asks, "Is there a scientific definition or equation for complexity?" I've got books on complexity, and I've been through them, and I'll be fucked if I can find that any clear definition for complexity. I'm still searching for an idea or a way to get my mind around what is the difference between a complex system, a simple system, and a completely random chaotic system, right? There's a lot of difficulties involved, but good question and something that we need to explore because
I'd probably think if we had Leo Filius on here, he'd probably talk about simplex and complexity based on was it what's that law that creationists always talk about, thermodynamics or thermo whatever?
so yeah,
He'd probably define it that way, like
the second law.
let's say
Yeah,
thermodynamics. That's it. So isn't that all about complexity?
yeah, and basically
Like
entropy
you're
and
basically in a system moving from
knowledge and
complexity to
information
simple.
and
Yeah, so he might define it that way in terms of physics. I'm guessing. Like I'm not a physicist at all.
well, I
That's
have
what.
a wonderful book I found. It's called oh god, I wish I see if I
But
can
did you
remember.
read it, Speed? That's the question that's thinking
No,
of
I
us.
have not read this book yet at all. But it's called I think it's called the second law. It's called Entropy for Children and
Dummies,
Concerned Parents or something. And
entropy.
he does a really good job of explaining entropy and information and all of that. I paged through it and it's something I want to read, but it's like a made simple book for entropy. And I'll see if I can find it and post it. But
Actually, speaking about entropy, I just finished that debate, Mr. Batman and RJ Downard, and
yeah,
one of the questions that was asked was, how do you define, like, how do you measure thermodynamics or the second law of thermodynamics? And
yeah,
he said, you'd measure it using Celsius and in some cases Fahrenheit.
that's what Batman said.
That's what Mr.
Oh
Batman
my
said.
god.
It made
Yeah,
me chuckle.
I dropped into right in the middle of the after show and they were talking about the second law of thermodynamics.
You measure it with temperature. That's the answer.
Batman was. And he is so full of shit. That guy is so full of shit. I can't believe it.
He managed to basically hold off till the end of the debate before he started accusing RJ of being a pedophile. So I was waiting for that. I thought he'd mentioned
oh
earlier.
God, I got to see that
But
part.
yeah, no, it was towards probably the last 15 minutes he started to accuse, and RJ he cracked it. He got upset, and so
Was
he
that
should.
was that in the Q&A session?
It was
We
in a
did.
Q&A session, and
Okay.
he basically, well, I think he said he didn't actually say you are a pedophile, but the way he kind of said it, RJ took it to mean he was accusing him as a pedophile. So
Right.
yeah,
Sorry,
it was
they
quite
went off on him, but
yeah, it was good old Mr. Batman in his typical
yep.
pedophile comments. Yeah,
Well, let's see. How's the canary doing?
yeah, well, he's still alive. That's good. Still alive.
Yep. Okay. I think I'm burnt out.
Yep. Well, it's what is it? It's past midnight in here.
Yeah.
Hey,
So
hey, look, just
takeaways.
no, no. Thanks for the time. So the first thing is thanks for the time. Thanks for explaining. I know it's a joke about the canary stuff, but it's kind of like a topic that I really am pleased to understand a little bit more about. I got to say, Speed, I've got quite a few questions in my head that I want to ask because I don't fully understand it. And all you've done is highlight even more stuff I don't understand. But at least I now know what I do and do not understand, and I could probably read more about it if I wanted to. So a big thanks for
Yeah.
that.
So let's see. So we might do a round two, but takeaways are transcription and translation.
Yep,
Learn
yep,
that.
I get that.
Figure out
Yep.
where that happens. Introns and exons. Understand that and how and the mRNA, and understand that junk DNA is bullshit, and the cell is a pile of mud. And we are going to continually learn new mechanisms continually, like 5 a day. And we're going to keep that is going to go on. And by the time we've learned them all, you know what's going to happen? We're going to evolve, and there are going to be new mechanisms that we don't know about yet because they're currently being created out there by life. And so this is never going to end. There's never going to be a point where you know genetics. Anybody. Nobody is going to know genetics. We're not capable. And by the time we catch up with life is going to outrun us again, over and over again. So, and the
We'll probably find
creationists are going to
something.
pull up paper after paper after paper to give us the exceptions to the rules. And
Yeah,
yet
but
they're the
I don't
ones
even
that
know why they
believe.
bother. I don't really understand why they bother. They can just pick any paper and just misrepresent it.
I know.
They don't need to find a paper that says it. They'll
Yeah.
just make it up.
Well, every time you bring up a paper to show them one thing, they're going to say, oh, here's a paper with an exception. But overall, they're the ones that believe that there are hard and fast rules.
Mm-hmm.
And all we have to do is show them that there are no fucking rules, that when they say junk DNA, that they're automatically talking bullshit. And we need to find a way to shut them the hell up.
Which I think is absolute evidence for evolution. That,
Yeah.
given the amount of time and the amount of, blinded, non-guided kind of evolution, you'd absolutely expect, things to be a mess, things to be jumbled, things to not make sense. You know what I mean? So things progressively getting more and more complex, but not in it, but in an unguided manner. In other words, it's very hard to make sense of it. So to me, this is just evidence of evolution.
Yeah. Yeah. The more complexity we find, the more it is sure that it was not intelligently designed, because intelligent design does not use complexity. It uses simplicity. So, but get one of them to admit that.
They'll never do it. Though at least you got them to admit Apple, APO, A1 Milano, as you're saying.
Yeah. Oh, and I have another one, the German cockroach, which at some point I could try to explain what happened with that, but that was an interesting one. That is new information.
Give a quick synopsis now. Well, a German cockroach.
Oh, the German cockroach. Oh, I can't. I can't do it quickly, and I couldn't do it justice right now.
Next time.
But basically, they evolved immunity to what the hell are they? Pyrethrins. They evolved immunity to an insecticide, and it was really cool how it happened. And it had to be something that you would call an additional information. Though I think creationists would find 3 ways to deny that, but
No, okay. I've
I'll
googled
get
it.
into it when I have more bandwidth. I'm running out, so I kind of want to end this, and we'll go back room.
Yeah, that sounds good.
Okay.
Well, thanks,
So
Scott. Thanks, Andreas.
yeah, and Andreas and Scott are the only ones in chat at this early hour. So we're going to end the broadcast. We'll see y'all later. Hope somebody learned something.
I certainly did. See you guys.
I think I did too.